Hello, I'm Bob Yauch, scientist in our molecular diagnostics group at Genentech. Welcome to Gene Expressions. Messages are sent between and within cells using networks of proteins called signaling pathways. One important signaling pathway is the MET pathway. Normally, the MET pathway is very active during the early stages of life, where it is involved in the development of the embryo. However, in adults, the MET pathway is much less active, but has specialized functions such as promoting the healing of cuts or wounds. The MET protein resides on the cell surface and acts as a receptor or a dock for another protein called hepatocyte growth factor, or HGF. When HGF binds to MET, it sets off a series of signals that tell cells to multiply and spread. The signals start when HGF binding brings two MET proteins together. This process, called dimerization, activates the MET proteins, which then activate other proteins inside of the cell. The signal is relayed from protein to protein until it ultimately reaches the cell nucleus, where the DNA is found. This causes genes to be turned on that tell the cell to grow, spread, and survive. When MET signaling is no longer required, MET and all the other proteins in the pathway are turned off until HGF binds to and activates MET again. Overactive and inappropriate MET signaling has been observed in a range of cancers, including lung, breast, colon, gastric, and liver tumors. In some types of cancers, overactive MET signaling has been associated with a worse prognosis. Additionally, overactive MET signaling may partially explain why some cancers worsen after initially responding to certain targeted therapies. For these cancers, it is believed that MET signaling can bypass the targeted signaling pathways, allowing cancer cells to survive again. But cancer cells inappropriately activate MET signaling in a variety of ways. They may have genetic mutations that cause the MET proteins to be activated even in the absence of HGF. Alternatively, cancer cells or nearby cells can generate too much HGF, which keeps the MET proteins on the cancer cells turned on. Another mechanism by which MET signaling can become overactive is through the production of an excess of the MET receptor on the cell surface. This is known as MET overexpression. With more than the usual amount of MET on the surface of cells, extra growth, spreading, and survival signals are sent to the nucleus. Cancers with overexpressed MET may be particularly dependent on this pathway in order to grow, spread, and survive. It is believed that determining the amount of MET receptor on tumor cells can be used as a biomarker to determine whether investigational medicines that target this pathway might be effective. This type of personalized strategy has been used to determine who may or may not benefit from other medicines. Researchers at Genentech have played a key role in improving our understanding of the MET signaling pathway and its relationship to cancer. At Genentech, we've been researching the MET pathway for more than 15 years. During the course of our research to target the MET pathway in cancer, we've developed novel types of investigational medicines. Thank you for joining me. I'm Bob Yauch. This has been Gene Expressions.